So my talk, exploring how environment and our genes impact colorectal cancer risk in Atlantic Canada. So previously, when I've given any sort of talk, particularly in the context of my research, I often start off with the question of, hopefully this works. Nope, it's not working. So let's see. Is this better? There you go. I often start off with asking the question, does anyone actually know what an epidemiologist is? However, obviously, in the world we're living in, that question's a bit moot. So instead, I'll just give a quick sort of explanation of who I am. So as Anne mentioned, I'm originally from Newfoundland, and for those of you who can see on our screen over here, well, that is St. John's. So I was born and raised in St. John's. Not surprisingly, I ended up completing my Bachelor of Science at Memorial University. I completed a dual degree in applied mathematics and biochemistry before heading off to UBC to do my master's. So for those of you who are familiar with Vancouver, you may notice Stanley Park here in the middle. Very beautiful area, really recommend it if you're ever visit Vancouver. And after my master's, I went off to do my PhD, in part because I really didn't want to get a job. Yeah. So I ended up doing my PhD with the School of Population and Public Health in conjunction with BC Cancer. So I spent roughly 10 years in Vancouver, of which pretty much 7 to 8 years was associated with BC Cancer, where I focused mainly on breast cancer. Not surprisingly, I looked at both genetic as well as environmental risk factors associated with breast cancer. And then shameless plug on the right-hand side here, or I guess technically stage left, these are my two daughters. This is actually a decently recent photo as we had just recently visited Vancouver. So they got to experience the area where my wife and I had lived for about 10 years. And unfortunately, well, not unfortunately, but if I stumble, apologies in advance. Unfortunately, my daughter was sick this weekend, so I spent most of it dealing with her fever as opposed to actually prepping for this talk. So bear with me. With that said then, because I spent the majority of my time in Vancouver focusing on breast cancer, given the fact that this is October, i.e. Breast Cancer Awareness Month, I thought it'd be really important that I at least address that. And being a statistician by training, well, numbers are obviously very important to me. So we have a bunch of numbers up here, and maybe some of them are shocking, maybe some not so much. The first one, number one, well, breast cancer is the most common cancer amongst Canadian women. Two, well, unfortunately, it is the second leading cause of death from cancer, right behind lung cancer. 25, well, a quarter of new cancer cases are breast cancer. So that makes up roughly 28,500 breast cancer cases every single year. And to put that into context, in some sense, hit it home, well, that basically equates to, on average, 78 women will be diagnosed with breast cancer every single year. Sorry, every single day. So to put that in context, when you get on a bus here in Halifax, that's basically 70, 80 people. That many people will be diagnosed every single day. 15, well, that is the number of Canadian women who pass away from breast cancer. And unfortunately, based on these numbers, well, then 1 in 8 women will experience breast cancer in their lifetime. And the reason why, again, I kind of bring this home is, again, breast cancer awareness, and that is why it's important then to talk about mammograms. In Canada, we have a great screening program. So for women who are over the age of 40, be it yourselves, mothers, sisters, aunts, well, if there's an opportunity there, please get your annual breast screening mammography. So with that said, moving on to the actual topic itself. So I'll give you a basic outline of what the actual talk will be about. We'll give an overview of colorectal cancer. We'll talk about some of the risk factors. For some of us, we talk about the idea of nature versus nurture. I'll then focus on specifically a current project that I'm working on that is funded by CIHR, as well as being supported by the Beatrice Hunter Cancer Research Institute. And then I'll talk about some of the challenges. And here, again, being a statistician, I'm going to highlight then the importance of math. And then finally, hopefully we'll have time, depending on where we are, for some questions. So diving into it then. Colorectal cancer, okay? So by the numbers. Unfortunately, it is one of the most common and the deadliest forms of cancer among Canadian men and women. So it is indiscriminate between the sexes. No, men technically have a higher risk for it, but it's still in terms of a non-sex specific cancer, one of the most common and unfortunately one of the most deadliest. In general, well, 24,000 Canadians will be diagnosed with CRC, so that makes about 10% of all the cases, and around 9,400 will die from CRC. Now, to put it in perspective, well, that basically means about 67 people will be diagnosed with CRC every single day. So a little bit less than breast cancer. However, on the flip side though, about 26 people will die from colorectal cancer every single day. So again, it is one of the deadliest forms of cancer out there. And so approximately then 1 in 16 Canadians, well, they will develop CRC in their lifetime. Unfortunately, 1 in 35 will die. So this gives you an idea of the impact that colorectal cancer can have on Canadians. And really, it's not that hard for us to kind of realize because a lot of us probably have been affected by cancer in some way, shape, or form. Myself, I've lost several uncles to cancer, same as my wife. So it has all touched us in some way, shape, or form. Now, in terms of the biology, obviously I'm sorry, I'm a biologist, but I won't dive too much into it. But CRC basically starts with our normal cells, okay, that can turn into polyps, or what we refer to as right here, because I got a big screen, adenomatosis, because I can never pronounce it, adenomatosis polyps. So basically, what we often refer as our adenomas. And so these are basically just all tissue growths, usually in the colon or the rectum. Unfortunately, some of them can actually become cancerous and eventually turn into adenocarcinomas. Now, overall though, things are on the upside, because if we look at the current trends, well, as we can see here over time, well, the number of new cases, well, they have been declining over time. And part of it though is simply a success due to, well, our screening programs, that they've been able to identify the cancer in the early stages. With that said then, approximately 47% of CRC cases, well then they are diagnosed in the early stages of development, what we often refer to as our stage 1 or 2. And so these will offer us the best possible outcomes. However, when we say only 47% are in the early stages, the downside is that means 53% are then in the later stages, where the risk of metastasis, i.e. the cancer spreading, becomes that much higher. So with that said then, part of the reason why this is something problematic is, well, if we look at some of the symptoms associated with colorectal cancer. Now, some of them are kind of obvious, such as, let's say, jaundice, so the yellowing of the skin. But some of them maybe not so much, such as, let's say, anemia, or maybe, let's say, weight loss. So these aren't signs or symptoms that are really of major concern or may not be as super obvious to us. With that said, though, some of the symptoms, they are a bit surprising. So some of the 5 most surprising ones, okay? Aside from blood and stool, well, again, trouble going to the bathroom, abdominal pain, discomfort, loss of appetite, weight loss, or maybe unexplained tiredness. These are, again, symptoms that really you wouldn't think of as big issues. A lot of us have 9-to-5 jobs. Some of us have very physical jobs, and as such, then being tired at the end of the day, at the end of the work week, maybe not having a proper diet, and that's why you've lost weight, well, they are pretty benign sounding. So you may not realize that this could be a sign of something a lot more significant. And so then our screening test, well, this is why they become so important to us, because screening test, it helps us find CRC before these symptoms actually develop. And as I mentioned before, when we catch it earlier, then the chances of a successful treatment, obviously they are going to be much better. And again, it lowers the risk of our cancer spreading to other regions. And again, when we look at the numbers, so we don't have to dive too much into what these numbers actually mean, but when we talk about when someone's diagnosed with cancer, this is a very common statistic we have, what we call our 5-year survival rate. And basically, the higher that number, the more likely you will survive. And as we can see here, for our early stages, our stages 1 and 2, for both colon as well as rectal cancer, basically what we're seeing here is you have a very good chance of survival if it's caught early. But unfortunately, when we move to our 3, when we move to our 4, right here, down there, number 4, there is less than a 15% chance of survival when we move to stage 4. This is when the cancer has spread to other sites, when there's risk of it becoming in, let's say, your lungs, and possibly your brain, or other regions where basically targeted therapy becomes that much more challenging. So with that said, This is my last plug for screening, but obviously screening is important. As we just mentioned, late diagnoses, well, unfortunately, means a worse possible outcome. So if you are 50 to 74 years old and you're not at high risk for colorectal cancer, then be part of these screening programs. The actual program itself, like the one from our Cancer Nova Scotia, is they literally will mail it to you. It is literally a little stick that you effectively poop onto or smear poop onto, you seal it up and you mail it back. It requires no real effort, but that one small action can help detect cancer in its early stages. So let's then talk about some of the risk factors. So the classic idea of nature versus nurture. Now, some of these things are what we refer to as being modifiable, such as, let's say, smoking. So we already know from beforehand that smoking is strongly associated with a lot of cancers, such as lung cancer. So it shouldn't be surprising that it is also associated then with colorectal cancer. But other things, such as what we often qualify as being non-modifiable, well, let's say your sex or your ethnicity, these are things that really you can't really change. Another way that we often classify, though, our risk factors is, again, to our nature versus our nurture. In other words, we can look at both the environmental as well as genetic factors. And one of the classic ones when we talk about genetic factors, let's talk about, say, family history. It's one that, again, we all, in some sense, kind of know about because we've heard of either ourselves having a family history or someone we know or care about having a family history. The thing, though, about colorectal cancer, though, it's a bit more complicated than that. Now, in general, CRC occurs, in terms of family history, we would say that CRC occurs in families more often than expected simply by chance. Now, importantly, despite no known associated genes. So emphasis here on no known, okay? We'll talk about that one later on in a second. But in general, individuals with a family history, well, they can have 2 to 3 times the risk based on the number of first-degree family relatives. So let's say if your brother, your parent, your kid, if they have, again, colorectal cancer, then that you now have a first-degree family member. The number of family members as well as their age will dictate your risk, okay? But now to make things a bit more confusing. So when we think about family history, then by default, we may often think about our genes. And so unfortunately with colorectal cancer, it's not quite like that because we also have hereditary or inherited types. And this is based on, well, basically those polyps I previously had mentioned. And so there are 2 major forms of this inherited one. One is known as familial, so our FAP version, and this is due to a mutation of what's called the APC gene, okay? Not super important what to know about, but the whole point is, unfortunately, if we have the APC mutation, then individuals with FAP, well, unfortunately, they have a much higher risk of colorectal cancer. In short, basically, individuals with this particular condition, they can literally develop hundreds to thousands of polyps in their colon. Unfortunately, if those are not treated, well, then they become malignant by the age of 40. In a similar vein, we also have what's known as Lynch syndrome. This was previously known as hereditary non-polyposis CRC. And again, it's a very familiar looking one. It ends up involving, again, the same type of polyps, but slightly different mutations. And the thing about this one here is, again, the risk is increased. The really interesting thing about it is Lynch syndrome can actually be associated or can be broken into 2 types. The type 1 is strictly associated with CRC, but unfortunately the type 2, that one is associated with breast cancer, colorectal cancer, bowel cancer, let's see, what else was there? Skin cancer, ureter cancer, bladder cancer, and a few other ones. So unfortunately, individuals with the type 2 Lynch syndrome, they had a higher risk of a lot of different types of cancer. So with that said though, although obviously these have very large effects, the majority of CRC cases or colorectal cancer cases are what we refer to as sporadic. So they're not directly associated with genes. So then they're often associated with things such as our environment, such as our physical inactivity or our sedentary lifestyle, smoking, and so forth. And so these factors are some of the more common ones that we have to deal with when it comes to, let's say, looking at the risk of colorectal cancer. And the one that I find most interesting is, again, smoking, we kind of see. Our diet, so diets high in meat or conversely very low in vegetables, that shouldn't be too surprising. But particularly for the younger group who are maybe watching, Alcohol. Alcohol is a very big risk factor, although it's had mixed reports, let's say, because some people talk about wine drinking and risk of breast cancer. The fact of the matter is, in generally speaking, and we'll dive more into that, alcohol is a very large risk factor for colorectal cancer. So, with that said, let's jump actually into my specific study. So this study is, again, being supported by CIHR, so a thank you to them, as well as the Beatrice Hunter Cancer Research Institute. And again, being a numbers guys, I have, in this case here, 4 important numbers that we'll be talking about in a second. And the first one is now our number 24,300. Now, for those of you who've got a good memory, well, we already talked about that number. That, again, is the number of cases of cancer we will expect across Canada in 2022. Now, importantly though, when we look at Atlantic Canada only makes about 10% of that population. And outside of, well, being a Newfoundlander myself, as well as obviously teaching in Nova Scotia, well, why is there so much emphasis on Atlantic Canada? And to give you context, although let's say Ontario obviously is going to be much, much larger, we do expect there to be more cases of cancer in Ontario. But obviously, we want to give you a number that allows us to compare, let's say, Ontario versus, let's say, the Atlantic region. And that's where our next number comes into play, 65. So, as I mentioned before, we expect more cases to occur in Ontario. And one of the numbers we use in epidemiology is known as a rate. So basically, how many cases per some set unit. So in this case here, the Canadian national average is somewhere around 65 cases per 100,000. So that gives you an idea of what the norm is in some sense for colorectal cancer in Canada. However, when we look at Atlantic Canada, well, this line down here, this line in red, well, that is the national average over the last, in this case here, 18 years. And this line all the way up here in green, well, that is Newfoundland, where I'm from. To give you context, on average, the rate that we expect in Newfoundland is somewhere near 110. So almost double the national average. Nova Scotia, in blue here, not that much better. We expect it to be somewhere around 90. Our purple and our green right here, our PEI and New Brunswick, well, we expect them to be somewhere around 80. Quite clearly, the top 4 spots for colorectal cancer belong to Atlantic Canada. So hopefully you're realizing why I'm interested in understanding the factors that affect colorectal cancer here, because for whatever reason, we are disproportionately affected by this disease. So the question is then, why? Why is it that Atlantic Canada is seeing these high numbers? Because again, when we look at some of the risk factors, well, again, sedentary lifestyle, okay? We do have, unfortunately, a very high rate of obesity in Atlantic Canada, okay? When we look at smoking, again, we have some of the highest rates of smoking. When we look at, again, diet, unfortunately, the majority of Atlantic Canadians do not meet the food recommendation guidelines for vegetables and fruit. And conversely, we have a lot more people eating red and processed meats, okay? And finally, our alcohol. So I previously mentioned that alcohol is a risk factor. And when I talk about this in my classes, it does surprise students when I tell them, hey, that drinking, that binge drinking, it unfortunately is a major risk factor, to the point where, well, as we may know, particularly in this region, alcohol is one of the most commonly used drugs. We don't have to go that far because, particularly for St. Effects, well, we just had our homecoming, probably St. Mary's, all the other universities in the area probably had their recent homecoming, and well, we've seen the aftermath. So we'll leave it at that. But the whole point here is alcohol can increase your risk of CRC by 60%. And importantly then, a proportion of Atlantic Canadians classified as heavy drinkers, it exceeds the national average. More than a third of Atlantic Canadians aged 18 to 34, and a quarter of those, 35 to 49, they're classified as heavy drinkers. So is it really then the environmental component playing a role? Maybe. But importantly though, none of these risk factors are unique to Atlantic Canada, right? We know that other regions, such as, let's say, Quebec, they have, again, a very high drinking as well as smoking rates. So if it's not then the environmental risk factor, maybe it's the people themselves. So looking around to the people physically in the audience here, how many of you here would say, know your lineage, for lack of a better term? Okay. So am I fair to say that probably you can trace your lineage to the British Isles?
Yes.
Okay, well, that's what Stats Canada says. This is only a few years old, but they actually surveyed people across the region, asking about, again, their ethnic or cultural origins. And we can see a very resounding theme here. The majority of Atlantic Canadians can trace their lineage English, Irish, Scottish, or basically the British Isles. So maybe it's the population itself, because when we think about the population itself, particularly how old the population is, well, a concept known as the founder effect may come into play. Because historically, well, unfortunately, Atlantic Canada doesn't have a really big rate of in-migration. We have plenty of out-migration, but given that a lot of people can trace their lineage to the first settlers, well, as we can see here, when we have one population migrate to, in this case here, a very isolated region, if they now, again, start to have more generations, well, the makeup of that genetic population may change. In this case here, we may see a founder population where some gene mutation now becomes very common within this particular region. And importantly, it's not hard for us to see evidence of this already. In this case here, so science lifts the veil on a deathly Newfoundland curse. For those of you who are from Newfoundland or maybe have been there or know people from Newfoundland, you may have heard something called the Newfoundland curse. And Newfoundland curse was associated with a gene mutation of called the AVRC. Don't need to dive into it, but the whole point is people, perfectly healthy, otherwise healthy individuals, they would drop dead in their 30s. And this was common. Based on current data, the prevalence, so how common this particular mutation is within the Newfoundland population is somewhere, I believe, don't quote me, somewhere between 1 in 5,000 to 1 in 5,000 to 1 in 10,000 Newfoundlanders have this particular condition. And again, not surprising that, let's go now to Quebec. In this case here, this individual, well, one of them, I'm gonna say this individual here, well, this person here had pancreatic cancer. Similar to colorectal cancer, pancreatic cancer has a very high mortality because like colorectal, well, the symptoms are very benign, something that you won't realize until it's much too late. However, a mutation that we found associated with an increased chance of survival of pancreatic cancer, well, it has traced its lineage back to the first French settlers. So the notion of a founder effect occurring within this region is not really, well, not that surprising. So that brings us then to my actual site. So for those of you who don't know, CanPath is known as the Canadian Partnership for Tomorrow's Health. And in this case here, CanPath is basically a longitudinal study involving right now around 330,000 Canadians that will be followed up for the next 30 to 40 years. There is so much data coming out of CanPath right now. And for me, I'm taking data from the BC Generation Project, where I am an affiliate researcher, as well as in the Atlantic Path. Atlantic Path, I need to give them a shout out because obviously outside of being a researcher with them, they currently have a Guinness World Record. And for those of you who are squeamish, sorry, but the Guinness World Record is for the most toenails. We have 25,000 samples of toenails right now at Atlantic Path. But the whole point is we have a longitudinal study that covers 330,000 Canadians for 30 to 40 years. And so we have questionnaire data. So I'll talk about, let's say, their personal medical history. We will follow them forward. So we'll eventually be able to see, let's say, new diseases develop. And as well, importantly, we have bile samples. So less disgusting than toenails, more typical, such as saliva or DNA. And so for me, then, that's where our next number comes into play. 820,000. So the nice thing about CanPath is that because of those bile samples, I now get a chance to look at approximately 820,000 genetic markers associated with every single one of these individuals. Now, most of them are based on what we refer to as a SNP, or a single nucleotide polymorphism. If you remember high school, the basic genetics, then we know there were 4 nucleotides. Our adenine, so in this case here, our cytosine and our guanine, they're being substituted out by a thymine and adenine, okay? So that is a single change in the nucleotide sequence. Now, it's important to realize though, that aside from the fact that this is the simplest one, because again, it's a single change, it's important to know that these are naturally occurring variations. They're not like the typical mutations we think about when it comes to cancer. And so importantly then, these SNPs, they can vary across both geographical locations and even vary within the same group of people with similar ethnic backgrounds. In other words, let's talk again about our founders effect. Because again, we may see certain mutations, let's say associated with the British Isles, but because of the isolation associated with Atlantic Canada, maybe that mutation has not had an opportunity to propagate within this region. So this now brings into focus of my research. So although we now know that there are genetic as well as environmental risk factors, what happens if it's a bit more? And so instead of saying, let's say nature versus nurture, well, instead it's nature and nurture. And so in this case here, this is referred to as what we know as a gene-environment interaction. So apologies to the biologists here. I know there is going to be a slight difference in how a biologist views an interaction versus a statistician, so bear with me for now. But the easiest way that I often talk about a gene-environment interaction is I use bathtub analogy, where we think of, let's say, an environmental exposure, such as, well, water leaking in or coming into this bathtub. And we think of our genetic component associated with, let's say, the size of the drainage pipe at the bottom. So in this case here, our first one here, if we look at this one, and what we mean in terms of visual, we can see here that there's really no difference between gene 1 and gene 2 with respect to our first exposure. So if we think of our first exposure, let's say as a leaky pipe, and our, let's say, our other genotype here, let's say, call that a small clog versus a normal one, we see that really there's no real difference based on that difference in genotype, which kind of makes sense. A small clog is not really going to make a difference with, let's say, a leaky pipe. But when obviously we turn that the faucet on, well, then we know we're going to increase our risk. Now, importantly, though, this is not an interaction because we see a same effect. Similar thing over here. Although we're seeing now a difference in our genes, the difference between our leaky versus our faucet are still within range. So the more important one then, right here, this is where we talk about the idea of an interaction, our departure from what we would expect. So one sort of analogy we can use is, let's say we have, again, a clogged pipe, but now instead of turning our faucet 1 or 2 times, we blast all the way. And so importantly, the combined effect or the overall effect is a lot larger than if we had looked at those 2 individual components. And one of the weirder ones then is the following here, where as we can see here, that risk in some sense has changed. Now, my analogy here doesn't quite work anymore, but a really great real world example is malaria. Particularly for the biologists, you probably know that, well, malaria in the context of sickle cell anemia, well, that has a protective effect. The individuals with that particular mutation for sickle cell anemia, well, they are a lower risk of the effects of malaria. So with that said, let's jump into now specific examples. And let's talk about alcohol. So in context of gene-environment interactions, we're going to be looking at particular genes, in this case here, ADL, so sorry, ADH, so alcohol dehydrogenase, as well as ALDH, so aldehyde dehydrogenase. And importantly, when people are exposed to alcohol, then they have to convert or metabolize the ethanol into, in this case here, acetaldehyde. And this is the problem one. Oddly enough, ethanol isn't really that bad in the beginning, but once we metabolize it into this one here, our acetaldehyde, this is the problem. This is the carcinogen. This is the one that, for example, can react to DNA to form adducts. And if we have adducts, then we risk, let's say, transcription errors, and that can lead then down the road to our tumors, okay? And importantly, we also have ALDH. So ALDH's job is to take that carcinogenic, that toxic aldehyde, and convert it into acetate, which eventually becomes, well, carbon dioxide and water. So importantly, we can see here, if let's say we have an individual who has, let's say, a SNP, a mutation, where let's say, for example, they overproduce, so their ADH is more active, then what happens if they're now producing a lot more acetaldehyde? Well, now more of that carcinogen is available to, let's say, form DNA adducts, again, leading to our tumors. Or alternatively, what happens if we have somebody who has a mutation in ALDH? Well, maybe now they can't get rid of our toxic carcinogen as quickly. So now we have a nice example of potentially a gene-environment interaction. Likewise, I just mentioned the role of our ALDH as well as our ADH. So this is another possibility of what we refer to as a gene-gene interaction. What happens if this is overactive? What happens if this is underactive? So now we're producing a lot more And again, we're producing a lot less or metabolizing a lot less of our toxic components. So that's the avenue that I'm exploring. Now, overall, there are a lot of different pathways that we can explore, particularly with the CAN-PAT data. One of them is polycyclic aromatic hydrocarbons. So in this case here, we often associate that with smoking, because again, unfortunately, we do have a high rate of smoking in Atlantic Canada. We have our, again, alcohol metabolism, and one that is surprising for a lot, well, circadian rhythm. In this case here, your sleep cycle actually can affect your risk of colorectal cancer. And finally, some DNA stabilizing genes. And one of my students over at Dalhousie, they're actually looking at the gut microbiome. Because again, when we consider the idea of your diet, how important your diet can be, well, that's another avenue of attack. Now, with that said, I'm just going to quickly talk about some of the preliminary results. So right off the bat, I have two data sets, one from BCGP, so that is again the BC Generation Project, and it involves roughly 1,300 participants. And later on, I'm going to have data from Atlantic Path, so roughly 1,250 participants. And unfortunately, due to delays associated with the pandemic, well, I haven't got access to them yet, so they'll be added in November. So the important note here, though, is that right now, the results you're seeing, they are based on, again, the BCGP. So importantly, any results that we actually see, it is possible that we still see the same effect, but it is also possible that it might disappear. So take anything you see here with a grain of salt. So alcohol consumption, again, we've already kind of established that we know is increased risk. And as we can see here, based on the data, well, we have elevated risk again, that individuals who drink 1 to 5 times a week, well, this number here, an odds ratio, we often equate odds ratio to risk. So basically double the risk, okay? And then if you drink 6 to 7 times a week, well, in this case here, we have almost triple the risk compared to those who are non-drinkers. Now, it's important to realize though, when we talk about interpretation of these numbers, although I just mentioned the word risk, the odds ratio itself actually isn't a risk, it's actually an odds. They're closely related to one another, but technically two different things. Another thing to keep in mind as well is the study design. And the study design will actually dictate, again, how do we interpret? Is it the risk of, in this case here, CRC amongst those who drink, or is the risk of having, let's say, an alcohol drinker among the cases versus controls? So there is technically a slight different interpretation. And the reason why I have to highlight this is the next one. So in this case here, ADH7. So this is one of the type of alcohol dehydrogenase genes, okay? Well, it was strongly associated with CRC, but in this case here, we have a ridiculously large number, almost 6,300. And so the important note here is I'm not saying that individuals with this mutation have 6,300 times the risk as someone who doesn't. So again, that interpretation is really important. At the end of the day, this simply telling us there is a very strong association. And the reason why is, again, how do we interpret these numbers? So when we look at here, we have our cases, the individual with colorectal cancer versus what we refer to as our controls, i.e. healthy individuals. For those of you who remember, well, I'm proud to ask that question, the high school genetics, TT-TC, so we have a homozygous group along with our hetero, and we have another homozygous group. In this case here, these are the homozygous major, or more specifically, the more common one, as opposed to those with, let's say, our TT, homozygous minor, those are the least common one. This is going to be then our risk group. So importantly, when we actually calculate an odds ratio, we're effectively timesing our top left by our bottom right, and we'll divide that by the product of our top right and our bottom left. In other words, 90 times 976 versus 2 times 70. So just from a simple math point, we can see why that number is really, really large. But again, it's not about risk, it's about likelihoods. And in this case here, based on the study type that I have, well, the way we interpret this is if we look at this, amongst the controls, basically everybody is the homozygous major, the CC, versus here, well, it's not quite evenly split, but there is obviously some favoritism towards having an RT. So we're saying then that our cases are roughly 6,300 times more likely the carriers of the T allele than, let's say, compared to our controls. And obviously that interpretation is very different from the idea of risk. So that's one reason why we have to make that important distinction. Another one, that really surprising one for some people, is the idea of sleep. So duration of sleep show that, in this case here, non-significant evidence, so not quite strong enough. It's telling us a direction, but it's not good enough that there is, or possibly some association. So this could, though, point towards, instead of duration, maybe the quality of sleep. Because for a lot of us, we probably have had scenarios where maybe you slept the standard 8 hours a day, but if you were tossing and turning the entire time, that quality of sleep may have decreased significantly. So maybe that's a different avenue. But importantly, in this case here, for one of my students, they looked at what's called the Cry1 gene, and this really regulates our circadian clock, so that sleep cycle. And unfortunately, is associated then with altered sleep patterns. And as we can see here, well, more realistic looking odds ratios. So in this case here, about 1.5 times. But again, we see a really ridiculously large one. Because again, when we look at a breakdown of genotype, we can see basically the exact same scenario. In this case here, again, we times 90 by 966, divide that by 1 times 11. So we have, again, that really over-accentuated value. But importantly, again, when we look at the breakdown, basically all the controls have the GG, and in this case here, basically all of the cases have our AA or AG genotype. So in this case here, we often refer to this as basically perfect classification, that your genotype can almost predict your, in this case here, case status. So this brings us then to some of the challenges that my team is facing, okay? And important to know is my team, because obviously any type of research, it's not a single individual. This is a group of individuals, and I'll highlight some of them later on, but 42 billion. So when we look at something like gene-gene interactions, so i.e. gene A, gene B interaction, gene A, B, and C interaction, then we refer to these often as higher order interactions. But really, this is just a math problem. So to give you context though, although I have access to 820,000 SNPs, if I were only to look at 1,000 of them, then if I were to look at all the order ones from 1 to 4, well, again, 1 to 4, so gene A, gene B, gene A, B, C, gene A, B, C, D, well, that produces 42 billion combinations. So as you can imagine then, trying to use all of data available, it's nearly impossible. And obviously one of the biggest problems then, well, is how do we actually deal with that? Because when we think about just the pure number, both in terms of computing power as well as computing time, it's not possible. Often when I talk about those type of numbers, if you were literally to say that you could do 1 million computations per second, so 1 million models per second, you still will not be able to cover all 42 billion within a few years' time. We're actually talking about a few hundred years' time. So again, going to all 820,000 is near impossible. Then we have what's known as a false positive. Now, for a lot of us, well, particularly in the context of COVID, we've heard of the idea of false positives and false negatives. Now, importantly, with a false positive, is basically thinking there is, let's say, oh, you tested positive on, let's say, COVID test, but in reality, you're healthy. We know that this can happen. This does exist. In the context of what I'm talking about here, though, is we may believe that there is, let's say, an association, when in reality, it was simply by random chance. Because importantly, when we talk about any of these type of studies in general, well, one of the biggest questions, particularly from a statistics side, is the sample representative of the population? And what I mean by that is, and I'll actually use the group here as an example. If I told you that I took a sample of 100 students, let's say here at St. Mary's, and I wanted to use them to make an inference about the average height of St. Mary's students. If I told you that the average height amongst my sample of 100 was 6'6", how would you feel about that inference? I'm now going to infer that the average height of everyone is 6'6". And presumably, all of you, as well as those of you who are online, you would say, that doesn't quite make sense. So let's give you context. Let's say by chance or by design, I end up sampling everyone from, let's say, the basketball and volleyball team. So now that number, it makes sense. But importantly, you have context. You understand the population. And clearly this population, or sorry, this sample does not represent the population. The problem in science, though, is more often than not, we will never know if our sample is actually representative of the population. So that's why then our false positives, we have to control for them. We have to acknowledge that there could be a chance that what we're seeing is completely by chance. And finally, as always with any type of science, money. It costs money. Right now, for my particular study, so I'm only actually genotyping approximately 500 people, so 500 people with colorectal cancer, and that's going to cost me around $35,000. So as you can imagine, then trying to, let's say, genotype all Canadians, it is going to be an astronomically large cost. So if anyone who is, let's say, online or here, maybe is associated with the Canadian Cancer Society, if you want to help support me, particularly for my next future directions, feel free to give me a shout. In terms of future directions, well, as I mentioned, I have data from Atlantic Path and BC Generations, but importantly, well, there are 7 cohorts in total. So we have Atlantic Alberta's Tomorrow Project. We have the newest ones, our Healthy Future Saskatchewan, as well as Cancer Care Manitoba. And then we have Ontario Health Study, as well as Cartagena. So at the end, if we can actually dive into using all of CanPath data, we have a much better chance of actually truly, again, understanding the associations between, let's say, some of the signals I'm seeing right now and whether or not it actually is associated with colorectal cancer. And again, it's important to realize that my data right now, based right now only on the BCGP, it is possible that the signals I see, it could disappear once I bring in the Atlantic Path. Even if it stands, once I bring in the other ones, again, it could possibly disappear. So that's why the more data we have, the better likelihood that we have a better understanding, a better idea of the true association between, in this case here, the genes of interest and colorectal cancer. So with that said then, as I mentioned before, it's a team effort. So some acknowledgements. So one of my former honor student who's now over at University of Toronto, Carly Feinstein, she did a lot of the work associated with our alcohol study. Alison Walsh, she is now a med student over at Memorial University, and her research in particular, it was supported by Research Nova Scotia, so a big thank you to them. Again, on Path, BC Generates Project, as well as CanPath, they're the ones who have put millions of dollars into making this data accessible, particularly for the researchers who do more quantitative stuff. There actually is a synthetic data set out there right now that you can apply for that basically mimics the real data. So it's a great opportunity to actually train with real health data. And finally, features Hunter Cancer Research Institute, as well as CIHR. So with that said, I think I'm somewhere around the ballpark of the 40 minutes. So then, obviously, thank you to everyone in attendance here in person as well as online. And then I'll leave this possibly for some questions, depending on if we have any.
Thank you.